It's my pleasure to introduce today's speaker, Dr. Lacey Hankin. Dr. Hankin serves as the Assistant Regional Ecologist for the Pacific Southwest region of the U.S. Forest Service, where the Regional Ecology Program provides expertise for sustainable science-based multiple-use land management. Dr. Hankin previously served as the fire ecologist at Yosemite National Park, where she supported science-based fire and resource management through fire effects monitoring and cross-disciplinary fire-related research. Dr. Hankin received her PhD in ecology, evolution, and conservation biology from the University of Nevada, Reno, and an MS in systems ecology from the University of Montana, where she worked on disturbance and climate impacts to tree regeneration and climate-informed seed selection. She's passionate about stewarding resilient disturbance impact ecosystems in the face of climate change through collaborative research and management. And with that, I'll turn it over to Lacey.
Thanks so much. And I appreciate the invite. Let me share my screen. Oops. All right, can you see the presentation?
Yep, everything looks right.
Great. All right. Hi, everyone. Thanks for joining. I'm really happy to be here. I've sat in on these webinars before. My name's Lacey Hankin. I am the Assistant Regional Ecologist for the Pacific Southwest region of the Forest Service, which essentially covers California. And today I'll be sharing some of the later work of my dissertation at University of Nevada, Reno on high elevation pine seedling climatic tolerances. And in this work, we tested ecophysiological limitations to seedling survival at the really early first growing season stage under an imposed drought and heat treatment. So first, I'd just like to thank my collaborators, Sarah Bisbing, Felipe Barros-Macias, and Ali Urza, as well as the many field assistants, greenhouse assistants, park and forest staff that made all the work possible, as well as thank you to my funders from University of Nevada, Reno, USDA, National Park Service, and California Native Plant Society. So I'm going to assume that at this point in the webinar series, many of you may have some background on these upper elevation 5-needle pine forests at this point, as well as the declines that have been ongoing for the last couple decades due to mountain pine beetle, drought, white pine blister rust, and altered fire regimes. So I'm going to abbreviate the background a little bit. But I do want to set the stage for plant-specific ability to deal with heat and water stress. And we know that climatic conditions are already exceeding seedlings' abilities to survive in some cases, as well as adults of the same species. And this is evidenced by widespread impacts of hotter droughts, which has implications for many ecosystem services like carbon storage, primary productivity, biodiversity, and many more. And high elevation forests in particular are snowpack dependent. And snowpack, especially in the Sierra Nevada, is expected to significantly decline, given that snowmelt timing is closely linked to peak soil moisture, which we can see in the figure on the left. Then these declines in snow and earlier snowmelt reduce available water throughout a warm growing season. Furthermore, peak soil moisture is expected to be even earlier in the season with climatic warming, which is shown across different mountain ranges in this figure from Parpold and Mullich. And you can see the impact of these different degrees of warming on the timing of peak soil moisture. So it's happening earlier and earlier in the growing season. So as we expect these declines in precipitation as snow, it's likely to accelerate the timing and rate of soil dry down, and that lengthens the period of growing season water stress. And this can be consequential for seedling establishment and survival. For example, some of our previous work highlights that soil moisture really is critical for seedling emergence in a planting context. And we found that soil moisture was the key driver of seedling emergence and abundance across numerous common garden studies that varied in soil, climatic conditions, topographic context. So it really was the underlying driver of early seedling success in the field. And we've observed this resulting pattern of soil moisture influence, but we wanted to explore more the underlying mechanisms of water-mediated plant survival and kind of what degree of stress these seedlings can withstand. And we know that water is essential for plant function. I think everyone knows that here, but water does move through the plants from the soil to the atmosphere along this gradient in water potential. And that's because the atmosphere is much drier than the soil. And water is lost through plant stomata during gas exchange, so photosynthesis. And it's a necessary consequence of photosynthesis, and it also provides a cooling mechanism for as the water moves off the leaf surface. And that's similar to how we sweat and evaporatively cool ourselves. So in order to conserve water in these water-stressed environments, plants can regulate those stomata and close them to prevent water loss. But as plants regulate those stomata, this process inhibits photosynthesis. So with prolonged periods of stomatal closure, plants may not be meeting their carbon demands for essential functions. And this is a hypothesized mortality mechanism known as carbon starvation. And it could be particularly important in these really young seedlings. But as water availability further declines, the tension in the plant increases, and then the plant is at risk of xylem cavitation and lethal hydraulic failure. And that's known as a widespread mechanism of plant mortality. But these two mechanisms can interact, so the higher the stress, the less likely the plant is to recover. So we would expect populations and species to differ in their sensitivity to these two mortality mechanisms and the amount of stress that they can withstand. So survival might depend on these species or population differences in drought-resistant traits. And at high elevations, these traits can be especially important for survival because seedlings are readily exposed to desiccation. They have shallow rooting depth. The strong winds and high vapor pressure deficits at high elevation can make them especially vulnerable to drying. So traits that increase access to water or reduce water loss are particularly advantageous. And these could include early investment in roots, conservative above ground growth, maintenance of plant water status, and a few more. So some of our previous work has shown that seed source climatic water deficit is associated with traits important for seedling survival. For example, in limber pine, we see that seedlings sourced from warmer, drier conditions have lower specific leaf area and specific root length. And these are strategies for water conservation. So there's, of course, this strong genetic component of trait expression, but we also know that plants can exhibit environmentally induced phenotypic change or plasticity, and this could also be an important mechanism for persisting under changing conditions. So to better understand possible mechanisms of mortality for high-5 pine seedlings, as well as the degree of drought and heat that these seedlings can tolerate, we asked, how do traits related to drought tolerance vary in response to different temperature and moisture conditions? And what are the ecophysiological limitations to survival during the first growing season? And we wanted to see if both of these things varied within and among populations and species. So this was intended as a lethal drought experiment, but we did expect that populations from warmer, drier conditions might possess traits that conferred greater drought resistance. So they might delay mortality or have reduced mortality rates. And we explored these questions with 3 dominant high elevation pine species in the Great Basin: Great Basin bristlecone pine, whitebark pine, and limber pine. And you're likely very familiar with whitebark and limber pine, but for those who might be less familiar with Great Basin bristlecone pine, it's a very range-restricted species in the Great Basin, and it occupies a very narrow elevational band as well. So while limber and whitebark have a really broad geographic distribution across the Western US and into Canada, we did address these questions just in the warmest, driest portion of the species ranges. So they really are occupying kind of the southwesternmost extent. We collected seed from 2 whitebark pine populations, 4 limber pine populations, and 4 Great Basin bristlecone pine populations. So we had 10 total populations across 6 mountain ranges. And the Great Basin is a super unique hydrologic unit, and it's characterized by these distinct basin and range topography that you can see in the photo on the right, where we have these really high elevation mountain ranges separated by broad arid valley bottoms. So because of this topography, forest populations in the Great Basin are particularly isolated to these sky islands. So to test seedling performance and survival under these drought and heat conditions, we looked at each seed source and exposed them to warmer temperatures and drought in paired indoor-outdoor common gardens at the Nevada Agricultural Experiment Station located in Reno, Nevada. So we planted seeds of the 10 populations across 6 treatments, and they varied in temperature and water conditions. The treatments included a well-watered control and 2 watering treatments that simulated 2 different timings of soil dry down. So those were separated by 2 weeks. And in the greenhouse, we set day and nighttime temperatures to the average growing season conditions from the source ranges. But due to a severe heat wave in Reno at the time, it ended up actually being a little bit of a warming treatment as well. So there was on average about 3 degrees Celsius warmer during the day. We then duplicated the same watering regimes outside where seedlings were exposed to natural temperature conditions. And these were well below the warm and dry limits of their ranges. So the temperatures were pretty extreme and averaged 8 degrees Celsius warmer during the day. But they actually had pretty similar nighttime temperatures to the natural range. So it was, they kind of experienced these huge temperature swings from day to night. So we summarized these moisture and temperature conditions 7 days following each treatment implementation, and then as well as prior to each mortality date. And in each treatment, we monitored growth and survival, measured water potential and leaf temperature, and then extracted seedlings to quantify functional traits. So although temperatures ended up being pretty extreme, our treatments did have, for the most part, the desired effect. So these are sensor data from each of the treatments. And so you can see air temperature, soil temperature, surface temperature, and soil moisture. And then the lighter colors are those outside, really hot treatments. And then the darker colors are the inside, warm treatments is what we're calling them. So you can see how much higher the outside treatment conditions, temperatures are. And you can also see in the soil moisture panel on the lower right that the how much longer it took for the soil to dry down inside the greenhouse. So there was really this critical interaction between the heat and the moisture conditions. So we had pretty high emergence for limber and bristlecone pine across all populations, but we had really low emergence in whitebark. And this was consistent with some direct seeding experiments we had done previously, as well as some other greenhouse common garden studies. So unfortunately, this limited some of the analyses we could do on white bark moving forward. So some of them you'll only see focused on limber and bristlecone pine. We saw pretty clear species level functional trait strategies, but no population differentiation. Rather, traits were primarily driven by treatment conditions. So they varied along those moisture and temperature gradients. And bristlecone pine exhibited the highest trait plasticity to different treatments, which was consistent with some previous findings where bristlecone pine exhibited the highest plasticity to different soil types. So for example, in the right here, we have trait distributions for carbon-13 composition and specific leaf area. And the vertical lines are the trait means for each treatment type. So in the outside hot drought treatments, we can see that traits shifted towards more conservative water use strategies, like lower specific leaf area and reduced carbon-13 discrimination. So lower specific leaf area minimizes that transpiration-driven water loss, heat load, and resists wilting. And then changes in carbon-13 discrimination are associated with increased water use efficiency. So seedlings were able to increase their water use efficiency in those outside drought treatments. We also saw some growth responses to the different treatments, but we thought these were more passive responses to changes in resource availability. So for mortality, we saw nonlinear responses to temperature and soil moisture extremes. So here in this panel figure, we have the number of seedlings that died per day according to soil moisture and then interacting with maximum air temperature. And we can see that there's this significant interaction. So daily seedling mortality increased significantly below a volumetric soil moisture of about 0.125, but this increase was only seen in the outside treatments where temperatures were a lot higher. So for moderate temperatures, which were the inside treatments, we could see that mortality was really low and kind of hovered around 0 the entire time. When we look at cumulative mortality, we also saw this nonlinear kind of threshold-like mortality response for all populations and species in those outside drought treatments. So in this panel figure, we have white bark on the left, moving to limber, and then bristlecone pine. Maximum surface temperature was on the right y-axis, and then cumulative mortality is on the left y-axis, and date on the x-axis. And we have similar coloring for each treatment type. And you can see, just to kind of start out, you can see that those warm inside treatments, mortality kind of hovered around 0 the whole time. I also want to note that the outside well-watered treatment also hovered around 0. But in those outside drought treatments, we can see that dramatic jump in mortality that didn't really differ in timing between the early and late drought treatments. Rather, it was likely caused by this short period of lethal surface temperatures. So pretty much all of the seedlings died between on a 3-day period between July 5th and 7th. And this period was associated with surface temperatures reaching almost 60 degrees Celsius. So that's definitely within the range of irreparable tissue damage. But I guess I just want to highlight, because I was really surprised by this, that those outside well-watered seedlings were experiencing the same surface temperatures, but they had really low mortality. So water really made the difference for surviving those severe temperatures. But we also wanted to kind of see what was going on inside the seedlings as they approached this mortality. So what was happening to their physiological function. So using a thermal infrared camera, we measured leaf temperature relative to the air, which we call delta T, And delta T can be used to infer whether a plant is transpiring. So thinking back to that water loss that occurs through the stomata and cools the plant. So when air temperature is greater than leaf temperature, this indicates evaporative cooling. And we saw abundant evidence of transpiration when water was available to seedlings. So similar mortality patterns, the relationship between delta T and soil moisture depended on air temperature. And this is shown in this figure for limber and bristlecone pine. So in blue, again, we have those lower air temperatures. And we can see there's a lot of variability in delta T regardless of soil moisture. So it's kind of all over the place, but in those hotter outside temperatures, which is shown in red, the need for cooling increases, but seedlings were only cooling themselves under higher soil moisture. So as soil moisture approached zero towards the left of each of those panels, the consequence of water loss is higher, and seedlings likely closed their stomata to prevent water loss. But this would result in an increase in leaf temperature, which can damage plant tissues. So if you think back to the carbon starvation hypothesis, this period of stomatal closure is where the plant is no longer photosynthesizing and not acquiring carbon. So it can lead to carbon deficits and a lower ability to deal with stress during this period. We also saw that in limber pine, seedlings were able to maintain water balance under a range of conditions. But again, as soil moisture declined under those higher air temperatures, water potential also declined, showing another nonlinear response as water stress increased. So while average water potential was not too severe, it wasn't indicative of a lot of stress. The lower values, kind of right as soil moisture is approaching zero, is within the range of documented growth inhibition and loss of hydraulic conductivity in some pine species. So we didn't directly measure hydraulic conductivity, but we suspect that these limber pine seedlings may have increased resistance to water loss in the roots and the needles, but likely only towards those temperature extremes. So the ability to maintain water balance under a wide range of soil moisture conditions by regulating stomata and resisting cavitation is a documented pattern in high elevation plants, and it could contribute to limber pine's broad geographic distribution. So it already occupies kind of a huge climatic range, and this could be one mechanism by which it can do that. So to wrap up, we found that the combined stressors of water limitation and extreme heat led to conservative water use strategies and declines in physiological function. So the joint stressors ultimately exceeded species tolerances and led to mortality across all species. But their ability to adjust trait strategies along the way and regulate functions to enhance drought resistance does provide kind of an intermediate mechanism for tolerating future conditions and uncertainties. And we didn't directly test hypotheses of hydraulic failure and carbon starvation, but our results do highlight a couple of potential mechanisms of seedling mortality. It definitely required the combined stressors of drought and lethal heat conditions, and together that caused complete episodic mortality. But heat stress alone, so in that well-watered outside treatment, had very little effect on seedling survival. So again, it all kind of comes back to soil moisture playing the dominant role in determining seedling outcomes. And despite vulnerability to extreme conditions, seedlings of all three species were tolerant of temperatures well outside their natural range of conditions. So on average, 3 to 8 degrees C warmer, given they had adequate soil moisture. So they may have wider margins for survival, but we are expecting prolonged periods of growing season moisture stress and declines in soil moisture. So we could still see some widespread impacts for seedlings of these high elevation five-needle pines. Great, so that is everything. I'm happy to take questions. I know that was a little short, but yeah, we'll have plenty of time to chat. Thanks.
Thank you so much for your talk, Lacey. Now, as Lacey mentioned, we're going to open the opportunity for our attendants to share your doubts, comments, and questions. We have plenty of time for them. So please type your questions or on the comments in the chat. And if we don't have enough time to go over all the questions, don't worry. Our team got you covered. We're going to send them along with your information to Lacey. So let's start with that. Okay, so our first question is as follows. Lacey, do you have any ideas as to why white bark emergence was low compared to other species? Was this a surprising result? And now that you have seen this result, does it lead to any other ideas?
That's a great question. And we kind of struggled with this over the 3 years of doing different work with these populations. When we tested it in the field with direct seeding, we planted it right before kind of the winter dormancy period to mimic natural, Clark's nutcracker dispersal. But we saw really low emergence that way as well. And then we did do some stratification for the greenhouse work, but it seemed to not quite be the right method either. So, and that was following, guidelines in seed handbooks and things like that. So I know there has been some work with different treatments of the seed coats and how that might increase emergence from seeds. So I think that might be worth exploring more, but just kind of doing the cold stratification, had pretty poor results. And yeah, I don't have a great answer for why it didn't work for us, even though we were following kind of established guidelines. I'd love to hear if anyone has more experience with it.
Oh, that's a great response, and it resembles to a lot of other results that we've experienced in other sides of the discipline around white bark and other white pines. That has to do along with like how do we implement experiments into species that like some methodologies hadn't been developed for it specifically. I wonder if anybody in our audience or yourself have heard or seen anything in other stone pines on the other side of the ocean. So if there are any other researchers in China or Russia studying this or like having similar issues. And we follow up with a question that kind of like ties up with these same limitations or methodological limitations, and has this research led you to other questions, or what do you see coming next, either spanning from these methodological limitations or just unexpected results?
Yeah, I think for me, coming from my current position where we're really thinking about kind of management solutions and how can we assist in recovery efforts of whitebark pines since the listing. I really want to dig into how can we make planting more successful. So because we saw such low success in our direct seeding work, and we're seeing a lot of vulnerability to desiccation and soil moisture, which is a big issue in these high elevation environments in the Sierra Nevada. What can we do feasibly in an operational context to have some planting work done? Because we're thinking through, genetic conservation as well as testing out different planting methods, if microclimate is making a big difference. And, that's all going to be big questions for, Forest Service recovery efforts in the next decade.
Yeah, that's actually really, really interesting. There was a a contention point during the conference this past year where there was a discussion about like if we're planting, what are we planting just with one seedling instead of multiples, since we're seeing this pattern naturally everywhere, and it has to do a lot with the duality of like whether putting more seedlings on a bigger area or trying to make it more efficient at a smaller scale. So it's kind of like a balance that we do need to find the best answer. And your question also, I mean, your answer also like takes me to think about Elizabeth Pansing's studies on how pilferage also could affect seeding. Or like how can we find a way that can increase the success rate of either planting or seeding directly? And I do believe that this branch of discipline is gonna take into rather insightful findings in the coming years. So that's pretty cool. We do have another question from John Bangandi, and it goes as, given that a recent study found that wiper produce larger and more resin ducts as a response to climate change, might this in any way help mitigate the effects of drought or heat?
That's a really interesting question, and I'll admit I think there are probably people on this call who have done a lot more work on resin ducts, so I'm not an expert on those. But I could imagine that you know, actually I think I'll just say I'm not sure. Yeah, I'm sure it I wonder if there's a kind of trade-off in terms of resistance to other stressors, not just climate, but beetle attack, things like that. But I don't know much about resin ducts, so that's
Yeah,
my unsatisfying answer. I'm sorry.
I do believe that is a great opportunity. This is perfect for researchers like Nick Kitches or Hannah Albertson to follow up on this area, maybe with you or any of our other attendants. We have another question from Elizabeth Barnes that goes as, are these results published yet? And if so, like which one of them are published or expecting to be published?
Yes, actually, all of our work is published. The specific study I presented on is, I think, Annals of Botany. I can post that link in the chat, and then our direct seeding work, as well as some natural regeneration work, has also been published on all of these same populations.
Perfect. We're gonna follow up with this and send it to our attendants after the talk. So our next question, or a couple of questions, come from Kelly Kerr. The first one is, were the foliar traits measured on cotyledons or true first leaves? And second, do the pressure chamber measurements reflect pre-dawn or midday time points? Wondering if differences between pre-dawn and midday might also support the idea of water use evaporative cooling.
Yes, so for the second one, it was midday stem water potential and also midday leaf temperature measurements. Yeah, I think it would have been interesting to do multiple time steps. We were just trying to work through a lot of seedlings with few people, and water potential measurements are time intensive. For the first one, it actually it depended a little bit on growth rates. So I would say a lot of them were not true first leaves, but some of Great Basin bristlecone in particular put out those first leaves really quickly. So some of that, we did capture some true first leaves in that.
That's right. How do you see these results being applicable for areas where it has been predicted that they're going to have more drought, like hot and dry areas for management?
Yeah, I think it really highlights, from our work that soil moisture is key. So I think, not this study, but our previous study showed that seed source can have a little bit of an effect in terms of more drought resistant traits. So you can consider seed source first, but then in a planting context for management, choosing those wetter microsites, even if you're in a drier environment. So, thinking about those shaded or topographic cues that would increase soil moisture, I think would be key in terms of successful survival.
Yeah, that, that is really interesting. So following up on this idea that this could like open up insight for multiple potential other sites where these climatic new conditions will arise or arouse. Um, what do you think could be specific or strategic areas where studies like this should be implemented in the future?
Yeah, that's a good question. I always really wanted to expand our reciprocal common garden studies beyond just the Great Basin, because we were working with these like the warmest, driest populations of these species. And so beyond just testing them within kind of you know the arid portion of the range, seeing if these are viable populations beyond, in the Rocky Mountains or Northwest. That was kind of a lofty interest of mine. Yeah, I think I got lost in my answer, so I can't remember your original question, but happy to expand on it if you
Yeah,
wanted
like
to.
yeah, absolutely. And I think we will have the perfect opportunity for that next September for the in the meeting in Bozeman, where I'm sure that many managers or people working with White Park, besides researchers, will be interested to expand on this. Because at least we do believe that is really interesting to know like that water availability does play such a big role in survival. So yes, and we do have one last question, which is, did you observe seedlings recover from the effects of drought even when it seemed like they were close to dying?
Um, we did see that. We didn't, so unfortunately we were using kind of mortality cues like wilting and browning. We weren't measuring gas exchange directly, so we couldn't tell exactly when a seedling died. But we definitely saw some recovery, of those kind of visual markers of approaching mortality, if they were watered. So, after the experiment was over, we kind of, you know, we were just kind of playing around with things, and we could see some recovery of those seedlings. But I think the ones in the drought treatments that approached essentially zero soil moisture, there certainly was no recovery of those. They died pretty quickly.
Interesting. Um, I'm gonna follow up with a couple of questions after a meeting with you that has to do with a recent with a presentation of a master thesis on White Park that has also to do with climate response. But for now, on behalf of the White Park Plant Ecosystem Foundation and the webinar organization team, I want to thank you again for your participation and be willing to join us. And for our audience, our next webinar will be on February 18th with Randy Moody from Canada, presenting his work on White Park pine restoration. What have we learned? Thanks again, everyone, for attending, and stay tuned for our upcoming installments. And if you do have any questions that you didn't post today, just let us know to our email, and we'll send it over to Lacey. Thank you so much for your attendance, and see you next month.
Thank you. Take care, everyone.